he was talking to us about this, the cellular response, the cell response. So we have this CAR T cell therapy, and it's called that because the scientists create a CAR, a chimeric antigen receptor, and they put it into T cells, and they use it for cancer therapy. And before we talk about the cellular response, let's just talk about the CAR, because it's just, students stumble on this concept. So they're like, we want to make a modified protein, the CAR. So how are we going to do that? And what's the answer?
We extract their T cells and put it in a test tube.
How are we going to build the protein itself? We got to make this protein.
By changing the regulatory region and the transcription unit.
That's true. How are we going to do that? And anybody can answer that. How do we get this job done? We want to make this altered protein, you know? And how do we get that protein altered?
Mutation.
What?
Mutation.
So it's actually not a mutation. It's something else that we're doing. But what's the work like that we actually do?
Well, we have to manipulate the DNA.
Yeah, so that's a key conceptual jump that surprises students. We want to manipulate a protein, make a different modified protein. How are we going to get that done? You know, people would say, oh, you must have little scissors and get that protein and get it pinned down and cut it and rebuild it, right? And the answer is no. We take the DNA, because every protein has to be synthesized, and it has a corresponding gene, and we take the gene and we manipulate the gene. We take the coding regions for these different genes, and we take parts of the coding region and we put the DNAs together and build one modified CAR gene. And then when we put that in the cell and the gene gets expressed, the ribosome just naturally synthesizes this altered protein. And so it's a conceptual thing, and I see over the years that students have a really hard time making that conceptual leap. We want to be fiddling with a protein. And why are we not fiddling with the protein? Because we're not. We never fiddle with the protein. We fiddle with the gene and that, you know, because that's how we would do it. Because it's really hard to grab a protein and cut it and paste it together. Nobody has the tools to do that. But we certainly have the tools to take apart pieces of DNA, put them together, match them up any way we want. And that's a little different than the concept we had all throughout our course, where you can just mix and match the regulatory region and the transcription unit. This is focused on, within the transcription unit, genes that have that code for proteins, and every protein has to have a gene that codes for it. They have, within the transcription unit, that coding region that got all the information on how to make that protein. And so we go to the DNA, because that's where the tools are to manipulate it. And that's the key thing, is that scientists have a lot of tools to manipulate DNA. And those are universal tools, because every cell on Earth has a DNA as its primary heritable molecule. So scientists really pressured that. Let's see, before you have that, I think Godwin maybe had a question? Or somebody, I thought, had their hand up. Oh, Hannah, yep.
Like in lab, is it kind of like what we're doing?
Just exactly, in the students that are in the laboratory classroom, that's what we're doing. We want to express this caterpillar protein, you know, and we're like, well, let's go to bacteria and put the caterpillar gene in bacteria, and we'll adjust it, the DNA, so we can get that misexpressed and actually have a caterpillar protein expressed in a bacteria. Yep.
And then you said that was different than what we're used to, which was...
Yeah, we're used to, in our course, introducing the idea that genes are made of these two parts, the regulatory region and the transcription unit. And we're used to either accidentally, through a chromosomal translocation, mixing those up, right? And getting like a misexpression of MYC or PDGF, right? We're really used to that kind of manipulation. And either it can happen by accident, or we could do that in a test tube. So we could take the myogenic regulatory region DNA and we can connect it to the GFP transcription unit from a jellyfish that glows, put that in, and mussels will glow like that. So that's our first level thing, and this is just a different strategy, and it's focused in on the coding regions and just mix and match those and put those together. And yeah, we're doing that in the lab, because in the lab we're building what's called a fusion protein, chimeric protein, because it's got part of the coding region is from bacterial protein, part of the coding region is from the caterpillar coding region, like that. So it's exactly the same thing. And that's a really important concept, how we build it. Then, today, and so we want to be able to go there, right? I think this is just a great thing that we think of Riley, and we think of Riley as somebody who has got this very specific interest. Want to treat this cancer. Going to do it using the principles that we're learning in our course, just all the principles. Apply all the principles, pull them all together and say, we're going to modify these T cells in ways that molecular and cellular biologists do and create this, this cellular therapy with this modified receptor that's going to get made, because we made a modified gene, put it in. Okay, so then the thing we're really talking about today are the cellular responses. Woo. Can you believe I still kind of have lost my voice? The cat went back to Boston on Sunday, and I used to have a cat. I had a cat that lived 26 years, and I was like this the whole time. And I was like, oh no, I love my cat, right? So today's the cellular responses. So what that's about is this T cell gets the gene to make a modified receptor, and when it bumps into a cancer cell, that receptor gets activated and the T cell does stuff. So that's the cellular response, right? And so we want to make a list of all the things we now know that the T cell does in response. So let's just, as our class, make a list of these are the cellular responses that CAR T cell makes. So what is one response that the CAR T cell makes? Oh, and I kind of lost my book. I came over here this morning, I grabbed my book, and it wasn't where I put it. So we have a silly sign-up sheet where you just have to put your name down. Yeah, so what does the CAR T cell do? Yep.
It's rearranging the cytoskeleton.
Yep. So one response is we have a change in cell shape. And you could say it hugs the cancer cell. It wraps around it. And what it's trying to do is it's trying to make more surface contact with it, because that's where the receptors are. And initially just a patch of them get activated, and that causes the cell change, and it does that very quickly. And as it wraps around, more of the CAR T cell receptors are going to be touching the cancer cell and connecting with the protein on the cancer cell surface. And that's called what's called a non-genomic response. And that means there doesn't need to be any new gene expression. It happens within a few minutes, and it should happen really fast. And so it's a good example of a non-genomic response, because it's like, we got to get this done, and we can't wait around for a transcription factor to recruit RNA polymerase and get new genes transcribed and get that RNA translated into a protein, and then have that protein do its job. So non-genomic responses can be very fast, like that. And so it's a good example of a non-genomic response. The only other non-genomic response that we've learned about in our course, so that would be a cellular response in response to some signaling molecule, is the response that cells make when they're activated by epinephrine or adrenaline. And that's a good one to learn because it has to be fast. And it takes a few minutes or longer to make a genomic response, to make new gene expression. So that's one, and it's one that Riley just told us about today. Now, that kind of response, non-genomic response, is a cellular response, and it would be a really beautiful example in this field of cell biology. Remember at the start of the course, I said our course, which is titled Molecular and Cellular Biology, is actually two courses together, molecular biology and cellular biology, which are actually different fields. They are overlapping, but they're really different fields. And our course has mostly been about molecular biology. Molecular biology is about gene expression and things that are involved in gene expression, so that spreads out because the transcription factors are regulated by signaling molecules and stuff like that. So molecular biology always starts out with DNA and genes, and then it just goes, okay, let's talk all about everything about the genes. Whereas cellular biology is things like the cell shape, and this actin is part of the cytoskeleton, the shape, the cell that gives it its shape. And that's a different sort of story. And so we don't have that many examples of these non-genomic responses because they're usually really pretty ones in cell biology. There are a couple chapters in our book back in section 3 that are really about cell biology, about the cell and what the cell is doing. And and we'll hope to get to a couple of those chapters. We'll get through chapter 20 and then we're looping back into our textbook. So that's a good one. Changing cell shape. It's good because cell biology is always, these are always just physical, visual, sensible things where you're like, okay, right? Like that, right? You're just gonna hug them and make sure that's the cell that you want, and then you're gonna kill it. Yep. Okay, so that's one, cellular response. Okay, what's another one that the cells make? Sorry? Yeah, so another thing that the cell is going to do, and we could kind of maybe make it sort of a set of things, and that is it's going to kill that cell. That's its thing. We're going to activate it, and that's its function. It's going to kill it, and there are several parts of that killing. And one of them is that it's going to release molecules that actually kill the cell. So if we drew it, here's the T cell, and it's going to release molecules like that. And they're going to bind to that cancer cell, and they're going to poke holes in its membrane, and then the granzymes that Eva mentioned are going to get inside the cancer cell and lead directly to its death. So these molecules, a lot of those are newly synthesized. So there's a lot of new gene expression. Like that. So that's one part of its killing, is I'm going to synthesize, I'm going to activate genes for molecules that are going to get released, and they're going to bind to the cancer cell and directly kill it. Yeah?
The molecules, that you talked about them, but should we know those specifically for the USM?
No. Yeah, like granzymes, because that's a lot of immunology. The signaling of the molecules that get released are generically called cytokines or chemokines. We would call them signaling factors in our course. They're a cell, this is like a source cell that's releasing these molecules and affecting a target cell. So we got a source cell, the T cell, affecting a target cell, a cancer cell. So we've learned a lot about this kind of stuff. But in this context, this is very much immunology, and there's special terms and details, not an immunology course. So we want to know that it's secreting factors and it's making them new, right? So, then up here probably we want to put that there's a lot of genomic response. And so the first thing up here we'd say is it's making a genomic response, and that's going to involve new gene expression. Including, we need to activate the genes that will be released from the T cell and bind to the cancer cell and kill it.
So would the WASP activation be step 4 through 6?
So the WASP is actually up here. So the WASP is activated here, and you don't need to know the detail of that word, WASP.
In step 2?
Yeah, but it's activated as part of this non-genomic response to say, let's hurry up and change the shape and really hug this cancer cell. And that will further activate it, the T cell. Get that? Yep. Yeah. And what you'll see, we'll talk we'll try to get to some cell biology and talk about cell shape some, but I think for now you would just say it changes its cell shape, and we don't need to get into that molecular regulation of WASP, or the cytoskeletal elements are called actin and actin, and we should learn some about that, but I think it's less important. Yes, Godwin.
So both the genomic and non-genomic responses, they are both new gene expression.
No, the key thing, a genomic response means the genome, and that means new gene expression. And that means regulating transcription factors. So there are, in the most general sense, a target cell can make two types of responses. Either activating transcription factors and making new gene expression, or not. And the not involves a lot of different things. Maybe activate that WASP, just a lot of different possibilities, but it's not new gene expression.
In the changing of the cell shape, you said it was non-genomic,
Uh-huh.
is that new gene expression?
Sorry, I hope I wrote that is a line there. So I wrote this up here first,
Yes.
and then this new gene expression fits with this one. So let me just put a line in there, because I hope I wrote those at different times and made that clear. That on our list, kill the cancer cell is going to involve new gene expression, because we want to release it. Oh, did I mess that up? So I should be separated. Non-genomic just means no new gene expression. Don't need to wait for new genes to be expressed. And then genomic response means got to get some new genes expressed. And we have a lot of examples of genomic responses. When we say, oh, we're going to activate MYC or ELK, those are all transcription factor stories, and those are new gene expression. We have to synthesize cyclins, right? Got to activate the gene. So that's genomic responses, and we've kind of become experts on that, right? So here the changing cell shape doesn't require transcription factors and transcription of genes, like that, okay? So this release of molecules would fit in that new gene expression, like that. So what are some other things that we should have? So it's getting messy already, but that's all right. So we have a good one, changing cell shape. Then we have another good one, kill the cancer cell. Oh, and let's add a little bit more to the kill the cancer cell. So one thing is that we're going to get new gene expression. We're going to synthesize killing factors that the T cell releases. And those killing factors are all like from immunology class. So we don't need to know the names of them, like that. Let me add another one here that we can sort of go like, make our list. This could be A, kill the cancer cell, release molecules and kill it. Then B, and Riley just mentioned this briefly, but another one is to release molecules that activate other immune cells. And that's, again, an immunology course concept. And that is that when the immune system responds, there are cells that help other cells, that help them go, hey, we got trouble, come here. So anytime you have an infection, and it's pink and warm and sore, it's because there's a lot of immune system cells in that neighborhood, and they're releasing molecules, and these molecules are what we would call paracrine factors, and they're just local, and they're encouraging other immune system cells to get focused and activated. So that's just important. Again, it's not an immunology course, but it's true that when the immune system gets activated, it's a bunch of cells, and they're all doing different jobs, including sometimes these other activated cells are showing up, and they're going to kill too. That's the big goal, is we want to get this thing, whatever this thing is, and it's hard to spot, right? It's a person's own cell, you know? We got to be careful here because we could cause an autoimmune disease if we're attacking the person. But it's something wrong with this cell, and we want to kill it. So there's a lot of checks and balances, and they include the immune system cells talking to each other and teaming up and saying, could you come help, you know, kill this person, or no, cell, sorry. Yes, it's cancer cell. Yes,
So,
sorry, yeah, I think Godwin and then Hannah.
for A, you said the first one is release molecule, and it also synthesize killing factors. Are those killing factors cytokines and chemokines?
Yes, yeah, but we don't need to dive into them because again, that's big time immunology stuff. And if you take an immunology, you know, you'll know already some of the stuff, and that's fine. We have plenty to deal with, you know, outside of immunology. So B would just be release other molecules that are going to be, again, the CAR T cell becomes a source cell. It's releasing molecules that are going to affect the cancer cell. It's releasing molecules that are going to affect other T cells. So that's a cellular response, right, is to make these. These other factors also need to be made. So another example of new gene expression. New gene expression to release molecules that are newly made that can kill this cancer cell, and others that are newly made that can attract other immune system cells, activate them, and have them help kill. Like that. Yep. Look for some more cancer cells. There's one. You guys look around here for some more like that. You know, it's a team effort, big time. That's a really famous thing. Again, not a course in immunology, but a good principle in molecular cell biology is, you know, cells work together. So that's a good one. So kill, right? Kill, and two kind of separate stories in the kill thing. So, so what we have so far is changing the cell shape, then killing, and within that we see new gene expression, so genomic responses like that. What's some more stuff that the CAR T cell does when the CAR activates? Yep, proliferates. And that's in our wheelhouse, right? We've been talking about that. And he says, oh, well, you're going to get June and Foss activated. So there's just going to be a really familiar path for us, like RAS gets activated like that. And downstream of that, we're going to activate something like ELK or maybe MYC, and we're going to get Foss made and June made, and they're going to team up and they're going to get cyclins made. So proliferation is a huge one. And we want more of these cells. So let's get them made. Also, we want these cells to stick around. So we want a kind of a memory, a kind of an immunity. So proliferation is a big one. And I think there's one more, and you know, I know this is a messy list, but I think there's one more that we should be able to say that the CAR T cell response does. And this proliferation, that would be new gene expression, and it's the kind of stuff that we're experts in. So activating transcription factors through signal transduction pathways like RAS and so forth. And I mean, there's more cell responses, but there, what's another thing that the cell does? And we've talked about this because it almost always goes hand in hand with when you tell a cell, hey, you should go through the cell cycle. What does the cell immediately start thinking and worrying about? Maybe I should just kill myself, go through apoptosis, because I don't want to become a problem. I don't want to become a cancer cell myself. So that the other one we want to learn out of this, and that's our next chapter, chapter 19, and that is that the cell is going to go through a response that's called anti-apoptosis. The signaling pathway is going to tell the cell, go through the cell cycle, and you're doing the right thing. We're going to inhibit apoptosis in the T cell because the T cell is worried. When it gets activated, it's like, okay, I don't want to mess up here. I could just go through apoptosis and eliminate myself. You know, I'm one of a trillion immune system cells. And I'm all of a sudden in the spotlight, and I'm worried about that. So it's really common that when cells are stimulated to proliferate, they also make a response of whether or not they should go through apoptosis. And sometimes, and it's one reason that we'll breeze through chapter 19, and that is because it's complicated, because we'll see there are some factors that inhibit apoptosis and some that activate apoptosis. And the cell is kind of actually doing both, because it's worrying, right? Sometimes when you're worrying, you're thinking of both things. I should go, I should not go, right? And so it's a balance, and it's just increasing the level of worry about the cell and whether it's in the right place and is doing the right thing or should just eliminate itself. Yep. Okay, so that's a good list. So it's a messy one, but first one we had is changing cell shape, and that's a great one. And we don't need to know the details of the proteins, but that it's a rapid non-genomic response where they're going to need more contact between the CAR T cell and the cancer cell, and that's going to mean more T cell receptor, more CAR T cell receptor activation. And then kill. And there's two parts of that. The CAR T cell releases signaling molecules and molecules that can kill the cancer cell directly, and it also releases molecules to encourage other T cells to come and get activated and also help kill that cell and any other cells in your body that are in trouble, like that. That involves new gene expression, like that. So we're going to activate some transcription factors, like that. Activate gene expression. Those genes get, transcribed. RNA gets translated in ribosomes. Those are proteins, and they get released from the cell. Now the third one is, I think, the proliferation. Classic. We need more of you guys. You guys are doing great, so we need to make more of you. We'd like you to stick around and keep helping. And so that's going to involve just a classic genomic response where we're going to activate transcription factors like ELK or MYC or June Foss, like that, and get that cyclin made or whatever it needs to be made to go through START, like that. The T cell's going to do that, like that. Just classic advancing through the cell cycle. And hand in hand with that, there's also going to activate these anti-apoptosis factors to say, no, good, you're good, keep going, you're doing a good job. Okay, anybody have any questions about that mess? But that'd be a good thing, a take-home thing, right, from the CAR T cell therapy thing is, what do these T cells do? And the answer is they bind to a cancer cell and they kill it. How do they do that? Well, they make a cellular response through that CAR, through that receptor, like that, right? So does people have questions or any things about that? Yep.
I don't have anything directed about this, but if somebody has something about this, they can go first.
Oh, yes. Any other questions? Yeah, and then we can talk about whatever Jeremy wants to talk about. Childhood pets.
No.
Okay.
I have a question about the CAR T cell, like making the CAR T cell.
Yep.
Because I think when I think about it, I think about like the estrogen response element having multiple domains, and so when you're making a CAR like receptor, how do you know that the genes you're inserting together are going to be like formatted in a way that works?
Yep. Yeah. And the answer is it's mostly trial and error. So we've seen in the lab course that undergraduates can build a chimeric gene. And so at these companies, they have many scientists at that level building these CARs and seeing if they're functional. Yeah, there's a little bit of protein biochemistry, people appreciating where you should try to join them together, but it's really kind of surprisingly modest. And so it's surprising how well the coding regions can be mixed and matched and create functional chimeric proteins out of it. Like that, where I'd say, like, let's take the arm of a human and glue it onto the body of a horse, and then on the other side of the horse, let's glue on the wing of a dove. And you're like, and all the arm and the wing are working, and you're like, yep. And you had to fiddle with it in lab, but we can fiddle with that stuff in lab really fast with a variety of different ways. Better put a little bit more of the wing on because it's not flapping like that. Yeah, it's really kind of surprising. It is very surprising to see how that it doesn't work out to put these little domain units in, like you mentioned, the domains of the estrogen receptor, and you could just mix and match them, even though, so it works out pretty well. Yes, other questions people have now about anything at all? Yep, and so then let's just go back to our stuff, start looking at some slides, this is where we're at. So, still following up on exam 1. That's just such an important thing, and if you aren't done with that, just let me know and we can walk through it. And my name is here. This week we're going to be reading chapter 19 because I thought we'd finish talking about chapter 18 today, but that took some time because we really want to absorb Rand O'Reilly's stuff. And again, you're not asked to read anything, so we want to figure out, well, what did Riley say that we need to know, right? And I'm like, well, what are the cellular responses like that? Also, we've talked before, his vast visit was the CAR, the gene, right? I ask about that, right? How would you make a chimeric protein? And the answer is you wouldn't fiddle with the protein. You'd fiddle with the coding regions of the genes, like we just talked about, like that. Yep. Okay, so then there's this thing about working on your term paper, and there's a description of that in the syllabus, and some of you are doing the 4-H STEM Ambassador Program and do not have to deal with the term paper, so it's just a subset of you, and I really encourage you, when you pick a receptor, send me an email, and that's just going to be happening outside of our class, and you and I can communicate about it, look at the syllabus. I just heard this morning from a student, and he said, I don't think you sent out a link for last Thursday recording. And I did. I just screwed up and that link sent out. So just before class, I sent out the link for last Thursday's class meeting, and I attached our syllabus again. It's on our Brightspace page, but there's the syllabus, and that's got the term paper information in it, and it just talks about what you need to complete for that. You could pick a receptor that we've talked about or one that we haven't talked about. You want to make sure it's a receptor, to make sure, that's not actually a receptor. Cyclins are not a receptor, like that. So it's very important to check in with me, email like that, okay? So that's working on that. Yes, Catherine.
In terms of exams, so we have exam 2 on Thursday the 20th.
Yep.
And then we have a week. Are we going to be covering any new material within that week, or is it going to be mostly reviewing?
Before the final?
Yeah, before the final.
Yeah. So we'll just keep talking about new stuff.
Okay.
Yep. Yeah. So we're now leaning into sort of a double exam thing, right? So we're going to have our second exam very near the end of the semester. We'll have a little bit more new material, and then our final. And so what you're learning now, you want to think about, could be asked twice, right? I mean, not twice, but I'll have a lot of time to ask you questions, ask you 100 questions about what we're learning now. 50 on the, second exam, 100 on the final. I'll try hard to make the second exam so it's more easy to manage it, but it will be just as hard. The questions will be the same type of questions. I'll just try to make them so that you don't have to spend as much time on some of the questions like that. And because I've talked about that a lot. So, but as far as just the challenge, so yeah, this material, and you can feel that, right? We learned some stuff in this course, and we're using that and adding to it in that context, and the process getting more familiar like that, because it's this looping thing. And a really important part of learning, I said at the start of the course, is recall. So you should say, okay, I'm going to tell the CAR T-cell story like that, How does that all work out, right? Like that, because I'll try to ask you questions about that, like that, and other things too. Yes?
One more question. Do you think we're going to have our second exams back before the final exam so we can look
Yes,
at them?
Okay. Yes. Absolutely. And I think it took me a little while to get the first exam back, but in almost all the classes I ever teach, I can get the exam back by the next class meeting. Yep. Yeah, so you definitely will have the second exam back, and I'll make a video of that too, like that, to help you with the final exam. Yep. Other questions people have? All right. So we talked a little bit about stuff, and let us just roll through a couple of things. So we've talked about chapter 18, except for this, we're going to talk a little bit about that. But we talked about the regulation of start, and we looped through that twice. And I would say, I would watch that. I would watch that class meeting from last Thursday again, because like we talked about retinoblastoma, like that, and like, people, it's just, the things, that we're covering are just fundamental concepts in animal biology. It's not human, it's not limited to animal, humans, but if you're interested in birds or whatever, it's the same kind of retinoblastoma thing all the time. And it's bound to this transcription regulation, right? Oh, gosh, and it's just so satisfying, right? Let's just scroll to, let's see what we have. Let's go get to that now, just to show you that. So let's just go this, and then we'll have a concept chat. So I'm just gonna put in this, and it says, so we've talked about start. We also went to the G2 to M transition, and we talked about that DNA damage checkpoint that involves the CDC25, removing inhibitory phosphates from another CDK. And now we're talking about start, and this would be an exam question like that. At start, the CDK gets activated. The CDKs are what some people call effectors, and it's a little bit of an unusual term, but it's kind of a good term. So CDKs are what we would call effectors, and that's to distinguish them from regulators. And this is a good concept just to learn those terms. So a regulator would be something like a cyclin. What is cyclin's function? Only to regulate CDK. Right, like that. What is CDC25's function? Oh, to regulate CDK, right? So there are regulators, like that. Transcription factors are regulators. And then effectors mean they're not regulating, they're doing the job. Like back to the CAR T cell, when it releases molecules that kill, those are effector molecules. And it's regulating the synthesis and release of those. It's like, I don't know, should I make these? But then it makes the thing, right? The actual thing. And the CDKs are the thing, right? And they do their kinases, so they phosphorylate targets. And so what is the thing? So what are the targets that we learned about? So these would be things that get phosphorylated at start. Can anybody think about one of those?
Sound like the nuclear envelope degradation.
That's a good one, but no. Okay, so let's just go with Joe's, because that happens at the start of mitosis, right?
Yeah.
So see, remember, like, we're talking about these different points in the cell cycle, right? And we went to 2 points, and they're different, right? And so Joe's one was a really good one, because I could also ask the question, what targets CDK phosphorylates at the start of mitosis? And that would include, phosphorylate the nuclear lamins, the proteins disassemble, the nucleus disassemble. That happens during mitosis. Another one we talked about is phosphorylate the histones. Histones rearrange, the chromosomes shorten and become visible, like that. So see, there are targets there that we want to know about, right, like that. And our book talked a little bit more about them, but so no, like that, but not to, to make Joe feel bad, because it's a really good answer, but not these, right? And it's important for us to distinguish those. Okay, so yes, Jeremy is going to say something.
The small inhibitory proteins, or CKIs?
Yeah, so we learned about that, this example of this P27, and when the CDK gets activated, it phosphorylates phosphorylates this P27, and it causes it to get destroyed. And this is an example of an effector where the CDK gets activated, and it phosphorylates this P27, and that degrades it, and it causes more CDKs to get activated. So this is an example of a positive feedback loop. Right? And it's this thing that, it's satisfying. Positive feedback loops are satisfying because you're like, let's gear it up. Let's ramp it up. So that's a good one, because we learned about small inhibitory proteins. They are just blocking, like that, right? Okay? So that's a good one. So that's one. P27, that's a target. And on your exam, right, I say, here I'm saying, what have we learned about it? So you just make a list on your exam. Boom, 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 like that. You don't have to explain them. But another question I could say is maybe, what is one target of the CDK kinase at start, and explain what it does. So there, your point, you'd be like, I picked P27, you got a point. Then I explain what it does, like that? Like that. So these are the, kind of questions that we have. The short answer questions, boom, boom, boom. So this one, probably like a 4-point question or something like that, because we have a few. So that's one. What is another?
P53.
Yeah, so it's not that though. Kind of, yep. So,
P21 targets
so Jeremy is just all in, and he said P53. And then Joe's all in, and he said P21. And, no, maybe you're right. Are you right?
Wait, I have in my notes that
No,
P53
it's
right
that-
now is
so-
P21.
Yeah, so the P53 is, if there's DNA damage,
Fair enough.
that is going to get phosphorylated, and then that's going to lead to the synthesis of P21. So, and that's good to bring up, because we have this little complicating factor. Remember, it starts a little bit complicated. We got the green light thing, just go, and that's what I like to call a control point. Let's get this thing going. But within that, then there's also a DNA damage checkpoint, and it's pretty simple. It's just DNA damage gets phosphorylation or activation of P53 by phosphorylation. It's a transcription vector binds activate P21. P21 is a small inhibitory phosphate protein, and it inhibits the CDK. So that's a good one to bring up. But again, no, friendly, teacher-student sort of communication. No. Okay. So sorry. All right. What's another one? Another one. What?
Retinoblastoma?
Yeah, retinoblastoma is just this. It's this thing where you're like, and we made a band. We made a band, a rock band, and our first album is called Retinoblastoma, right? Because it is just nuts, and it's going to get phosphorylated. And I think before we dive into it a little bit, and maybe we just want to have a concept chat today, but before we dive into retinoblastoma a little bit, I'm going to say, I think there's only one other target that I think we learned about, and what is it? EVA.
MCM?
MCM. So I think this list goes P27, retinoblastoma, and MCM for our course. And MCM are those proteins that help DNA polymerase find where to start copying the DNA. And so just appreciate that our list is intended to be a minimal list to demonstrate different actions that are happening, right? The first one, P27, we got to get this ball rolling. So let's get some more of our team out on the field. There's some CDK that still have inhibitory P27 bound to them. Let's phosphorylate P27 and get that out of the way. Then
Last week we said the 5 examples of the start were activation of target genes for cyclins like MYC, June, FOS, retinoblastomas, and then
sorry, go through them one at a time. So last time we said, what are the actions at start?
activation of target genes
Okay, so
for cyclins, which was MYC, June, and FOS.
yes, but okay, and so that happens, but this is like the smaller
Yeah.
story within that, because that is a story about how we need to make cyclins
Right.
to activate this.
Right.
So
I'm
yes,
just saying
but that's one. But so that's one. Yeah, that you have. What's another thing you have?
retinoblastoma.
Yep. So we'll talk more about that. Direct effector.
Yeah, and then we had the 3 P27, P53, P21.
Yes. Yes, and we talked all about those, about how the P53, P21 is a different story that is not about turning the light green. It's about school patrol and getting them out there or not, because it's DNA damage checkpoint.
Yep.
And so, yes, so that's kind of a bigger picture of we got to get this thing activated because this is what's going to happen.
Okay.
Well, this is what we need to get it activated, and then this is what it does when it's activated.
Gotcha.
So once it's activated, we get a further positive amplification or activation. Then we'll talk about retinoblastoma, then MCM. So those are, I think, the three we learned about. So we want to be solid on those. And it's a good thing, right? And this is kind of like a review, like that. But I just don't spend much time reviewing because I'm like, oh man, what? Like we've read like 5 chapters in our book, right? I just am like, let's try to see if you have questions, and if not, we'll just keep moving on like that, right? But that's because the kind of studying you do is a reviewing thing, right? Like that. And we could meet, or you could talk to Riley about it, or classmates. But it's this thing where you reflect and rebuild these kind of notes, right? To kind of like, okay, what's the bottom line? Because we talk, and then if the exams are just like, what's that? What's that? What's that? And the exams have these kind of formats. So you're studying, the effort is to arrange things in a way that you're like, I'm preparing questions like that I can answer. Yep. Let's see, somebody had their hand up about something. Yep, Hannah?
So all of these are listed on regulators.
All these are regulators. That's a good question. So when we say CDK is an effector, it's what are its effects? What's it doing? And so we got that it's going to help activate itself. So it's kind of circular. But then we got this retinoblastoma, and it does other things like this. But retinoblastoma is a really satisfying example, so we'll talk about it. And then it does things like phosphorylate MCM, like that, okay? So let's just go back and look again at this retinoblastoma. Not because, like, we should or something, but just to sort of underscore, the stuff we're talking about is just in class, it just goes boom, 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 because it's just fact, like this. So you just look at this thing, right? And the first thing is, okay, it's transcription regulation, and we're just so solid on that because we started our course with that, right? So target genes, response genes to that transcription factor, which is E2F. So that's the first thing to say. E2F is going to activate gene expression. And what kind of gene expression? And the list is just so satisfying because when the light turns green, we're going into S phase. What do we need to make? We need to make DNA polymerase. There's some around, but we need a lot. And it's a protein, so there must be a DNA polymerase gene, right? And we're just going to crank it up, big time, like that. Then we need to make the nucleotides, AGCT, because the DNA polymerase is going to be grabbing those and cranking down these giant chromosomes that humans have, right? And then we need to make more MCM proteins. There's some around, and CDK is phosphorylating them and activating them. We got to ramp this up. It's a factory effort, and it's a bunch of new gene expression. So that's E2F, right? It's a crazy transcription factor that sits at the crossroads of turning the light green, right? I mean, it's like it's going to provide the light bulb and the green filter and the electricity to make this show go like that, right? It's just that direct. And so what's retinoblastoma doing? It's saying, nope, it's a tumor suppressor. It's just like, no, we're not going to, no, we're not in S phase. We're in G1, folks, and we're waiting. And then CDK gets activated. Chemical reaction is it phosphorylates retinoblastoma releases it. So retinoblastoma is a transcription factor. It's a co-repressor of E2F. And it's just a way to keep that show shut down. It's just cool, that's all. And that's why it should be the title of our first CD, album, whatever, on Spotify, and it'd just be like crunching through it, because it's that satisfying. Okay, so we didn't have a concept chat, and we also have to stop. So I have this funny sign-up sheet, just autograph your name. I hope I can find my book. If not, our course is over, and I'll send